Good afternoon, all of you. In last lecture, I had discussed the evaluation of microbial stability of formulation. Today, we will see the next point of our syllabus, that is animal cell culture. We will see first introduction about this. So, what is cell culture? The cell culture refers to a process by which it is the process by which the cells are grown in a controlled artificial environment. Yes, what is cell culture? It is the process by which the cells are grown in a controlled artificial environment. The cells can be maintained in vitro outside their original body by this process, which is outside the body by the use of this process, which is quite simple compared to organ and tissue culture. Yes, this cell culture is simple as compared to organ culture and tissue culture. In a cell culture technique, cells are removed. Yes, in the cell culture technique, cells are removed from an animal or the plant and grown subsequently in a favorable environment. Yes, in cell culture technique, the cells are removed. Cells are removed from an animal or the plant, and these are grown in a subsequently in a favorable condition or the favorable environment. For animal cell culture, the cells are taken from an organ. For animal cell culture, cells are taken from an organ of an experimental animal. Then the cells may be removed directly or by mechanical or the enzymatic action. Yes, cells are removed by whether first that is directly, or second then mechanical, or the third that is enzymatic action. The cells can also be obtained previously made cell line or cell strain. Then examples of cells used to culture are fibroblast, lymphocytes, then cells from cardiac tissues, then cells from skeletal tissues, then cells from liver, cells from breast, then skin, and cells from kidney, and also different types of tumor cell. So this is the introductory part about cell culture. Next, that is types of animal cell culture. Types of animal cell culture. Based upon the number of cell division, the cell culture can be classified as a primary cell culture. Yes, based upon what? Based upon the number of cell division. The culture, based upon number of cell division, the cell culture can be classified as a primary cell culture, organ culture, tissue culture, and cell lines. Then cell lines can undergo finite or infinite cell division. First, animal cell culture. In that, primary cell culture and second that is secondary cell culture and cell lines. First, that is primary cell culture. So this is the cell culture which is obtained straight from the cells of a host tissue. Yes, this is the cell culture which is obtained straight from a cell of host tissue. The cells dissociated from the parental tissues which are grown on a suitable container, and the culture thus obtained is called as a primary cell culture. So, such culture compromises mostly heterogeneous cell, and most of the cell divide only for a limited time. However, these cells are much similar to their parents. Example of primary cell culture: if liver cell are desired to be cultured, if the liver cell if the liver cells are desired to be cultured, the same growth conditions of the liver in vivo need to be replicated in vitro. And the primary cell culture is of two types, that is adherent cell and suspend. Adherent cell, first, these are the cell which encourage the dependent and the propagate as a monolayer. Then these cells are need to be attached to a solid or the semi-solid substrate for a proliferation. It means that these cells needs to attach to a solid or the semi-solid substrate for proliferation. Then these adhere to the culture vessel with use of an extracellular matrix, which is generally derived from tissues of organ that are immobile and embedded in a network of connective tissues. Then Fibroblasts and epithelial cells are of such types. When the bottom of the culture vessel is covered with a continuous layer of cell, when the bottom of culture vessel is covered with a continuous layer of cells, usually one cell in thickness, these are known as monolayer culture, and the majority of continuous cells grow as a monolayers. As being single layer, such cells can be transferred directly to a cover slip to examine under microscope. Then second, that is suspension cells. Suspension cells do not attach to the surface. Suspension cells do not attach to the surface of the culture vessel. These cells are also called as anchorage independent or the non-adherent cell. Yes, these cells are called as non-adherent cells, which can be grown floating in the culture medium. Then hematopoietic stem cells, which are derived from blood, spleen and the bone marrow. Yes, the hematopoietic stem cells, these cells which are derived from blood, spleen and the bone marrow. And the tumor cells can be grown in a suspension. These cells grow much faster. Which do not require the frequent replacement of the medium, and it can be easily maintained. These are homogeneous types, and the enzyme treatment is not required for the dissociation of cells, and similarly this culture have short lag period. Then third, that is confluent culture and the necessity of subculture. The cells are isolated from tissues and proliferated under the appropriate conditions. They occupy all the available substrate, that is reach confluence. Then for few days it can become too crowded for the container, and this can be detrimental to their growth, generally leading to cell death if left for long time. Then the cells thus have to be subcultured, that is portion of cell is transferred to a new vessel. That is the portion of cells is transferred into a new vessels with fresh growth medium which provides more space and nutrients for continual growth of both portion of cells. Hence the subculture keep the cell in healthy and in a growing state. Then next, secondary cell culture and cell line. When the primary culture is subcultured, it is known as secondary culture of the cell line or the subclone. I am repeating here, when the primary culture is subcultured, then it is known as secondary culture of the cell line, or it is called as subclone. The process involves removing the growth media and dissociating the adherent cell. Usually it is enzymatically. Then subculturing of primary cells to different divisions. The subculturing of primary cells to different divisions leads to a generation of cell lines. Then during the passage, the cell with highest growth capacity, the predominate, resulting in a degree of genotypic and the phenotypic uniformity in the population. However, as they are subcultured serially, they become different from the original cells. Then next classification on the basis of lifespan of culture. The cell lines are categorized as first, finite cell line, and second, that is continuous cell line. Yes, this classification which is based upon lifespan of culture. First, that is finite cell line, and second finite cell line, second that is continuous cell line. In finite, first that is finite cell line, the cell lines which go through a limited number of cell division having a limited lifespan and which is known as finite cell line. See here, I am repeating, the cell line which go through the limited number of cell divisions having a limited lifespan and which is known as finite cell lines. Then cells passage several times and then lose their ability to proliferate, which is genetically determined event known as senescence. Then cell lines which is derived from primary culture of normal cells are finite cell line. Next, that is continuous cell lines. When the finite cell line undergo the transformation, and acquires the ability to divide indefinitely, it becomes a continuous cell line. Then such transformation or the mutation can occur spontaneously. Or it can be chemically or the virally induced or from the establishment of cell cultures from the malignant tissues. Then the cell cultures prepared in this way can be subcultured and grown indefinitely as a permanent cell line. It is and are immortal. Then the cells are less adherent, then it is fast growing, then it is less fastidious in their nutritional environment and which are able to grow up to the higher level density and different phenotypes from the original tissue. Then such cells grow more in suspension. They also have the tendency to grow on the top of each other in multi-layers on the culture vessel surfaces. Then we'll see the advantages of animal cell culture and disadvantages of animal cell culture. So what are the advantages of this animal cell culture? First advantage, it provides controlled physiochemical environment like pH, temperature, osmotic pressure and oxygen. Then second, it provides controlled and defined physiological conditions. It provides controlled and defined physiological conditions. Then third, it achieves homogeneity of cell types through serial passages. Then it requires smaller quantities of reagent as compared that of which are required for in vivo, and thus it is economical. Then it avoids legal, moral and ethical questions of animal experimentation. Then it avoids legal, moral and ethical questions of animal experimentation. So these are the advantages of animal cell culture. Next one, that is disadvantages of animal cell culture. First disadvantage, it demands expert for interpreting and controlling the behavior of cells in culture. Then second disadvantage, it is 10 times more expensive. It is 10 times more expensive for some quantity of animal tissue. And third disadvantage, that is it has unstable unapplied chromosomes. So this is all about the animal cell culture. Next part we will see in next lecture.